So welcome everybody to our December BMS Talk. And it's my pleasure, first of all, to introduce our chair for this evening. Linda Seward spent her career in magazine and book publishing with a focus on quilt making. During the COVID lockdowns, Linda began to study the natural world in earnest, photographing and researching everything she found on daily walks. She's now totally immersed in mycology, involved in her local Oxfordshire fungus group, and writes articles about fungi for regional newspapers and magazines. So thank you very much, Linda, for being with us this evening to chair, and I'll hand over to you now.
Thank you very much, Sally. So welcome, everybody. I'm really excited about this talk tonight because I've actually had a sneak preview and it's wonderful. I'd like to introduce Cameron Diekonigin. He's the chair of the Edinburgh and Lothians Fungus Enthusiasts, ELFE, and he's a part-time amateur mycologist who enjoys identifying microfungi, particularly members of the obsolete Pyrenomycetes, that's black Ascomycota, that are most easily found on fallen wood and dung. Although he grew up in the southwest of England, Cameron currently lives north of Edinburgh, over the Firth of Forth, in Kinross-shire, where there are many species of black fungi growing on sticks. And I will now hand over to Cameron, and then you'll see me again at the very end.
Thank you, Linda. So yes, black things on sticks, an introduction to lignicolous melanised fungi. Just to let you know, this talk will last about 50 minutes. So the first thing I would like to take you through is what we're going to cover in this talk. So who is this talk aimed at? The benefits of collecting black things on sticks. What are they? What are they not? Identifying the fungal groups and the terminology used. We're going to key out a fungus together. We're going to do it live on the screen. And then I'm going to look through some common species as well as some highlights. And then I will wrap up at the end with resources for study and some of the challenges if you wish to take up looking at these fungi. Before we do any of that, though, I would like you to get involved in this talk. What you're going to see is a series of photos, such as the one that you can see on the left just there. And for those that aren't named, I'd like you to make a note of those that you know. Towards the end of the talk, I'd like to use chat and let us know how many of the 38 that you knew of. So for example, Mark here, he knew 6 out of the 38. I will remind you at the end when to send in your numbers, and there is a prize. Next time we meet, I will award you with a stick with a black fungus on it. So yeah, please do get involved and let us know how many of the fungi you knew of. Okay, so the first part I'm going to look at is who this talk is aimed at. So it could be you are currently interested in the larger guild fungi and some of the larger Ascomycota, scarlet elf cup, green elf cup, and so on. So you've already got a bit of an interest in the larger fungi. It could be you have a microscope to look at spores to aid identification of some fungi. You might be someone who doesn't even know where to begin with the small fungi, the microfungi, or perhaps you didn't even know they existed. Or you would like to fungus foray outside of the main autumn season, but you don't think there's much to find at that time of year. So if you tick one or any of these boxes, I think you might be interested in this talk. So let's have a look at the benefits of collecting black things on sticks. I've got 9 for you. The first one is that many of the species are easier to identify than you would expect. They are literally everywhere that you can find sticks, so they're really easy to come across, and you can find them throughout the year, and they're easiest in the winter and the spring, partly because a lot of the other stuff is no longer around, and also a lot of the wood is easily visible. And here is a photograph of the Edinburgh and Lothians fungus enthusiasts out in reasonably heavy snow, and on that day we had a really good haul of records, of which a fair number of them were black things on sticks. There is a taxonomic bias within fungi, and we tend to go for the larger fungi and the more dramatic looking ones. And if you look at or watch the last BMS talk by Christopher Ellis, he touches on this. There will be the almost overnight geek effect. People who go around collecting sticks with black things on do stand out from the crowd. Many are amazing to look at, particularly under magnification. And one single stick or leaf can provide up to 5 or 6 species, so you don't need many sticks to get a really good number of fungi. There are some really rare species that actually just need someone to go out and look for them, so you can contribute very quickly to the records. And finally, the fruit bodies, and let's call them fruit bodies for the purpose of this talk, can last for weeks to months. And the great thing about this is that once you've collected them, not only can they survive desiccation, they can survive our busy lifestyle. So you can go and ID them at a time that's convenient for you. So 9 really good reasons to go out collecting these fungi. So the question is, what are black things on sticks? The first thing that I would like to highlight is they're predominantly in Ascomycota, and this is the fungi that produce spores inside an ascus, a long tube. There's usually 8 spores in the tube, and they fire them out of the top into air current. So they're predominantly in this group. The key features of the black things on sticks fungi are as follows. They're lignicolous, so they grow on dead wood, often still attached to the tree. Because of this, because of their exposed position, they're often melanised, they're black, and the melanin helps to protect the genetic contents from ultraviolet light. But because they grow in exposed positions on wood, they're also xerophilic. They're able to survive very dry conditions, desiccation on the exposed wood. To give you an example of how good they are, this is an example of one of the fungi, and I've sliced the top off, and you can see inside the little round fruit bodies, and they look hollow. What I had done is I've sprayed this with water, and then 30 seconds later, that's what the fungus now looks like. So within 30 seconds, it absorbs water, and the asci with the spores in are ready to start discharging in a very short space of time. So they're really well adapted to the extreme conditions on wood. They're persistent. So the fruit bodies may be perennial, but they certainly last for several months. And even when dead, spores can be retrieved. So again, you can collect them today, and you can pop them aside for a couple of days to a couple of weeks, and then come back and ID when you've got more time. And finally, they produce lots of fruit bodies, and this is really good. If you are quite new to this group and you want to learn and you want to practice, lots of fruit bodies means that you can practice your slide preparation and get the skills right with many fruit bodies. So what are not black things on sticks? So I've got 6 examples for you, all of which have been brought to me on forays in the past, and I've been asked whether they are fungi or not. So burnt wood, bird droppings, particularly the dark centre, bootlaces, so these are the rhizomorphs of honey fungus, paint splashes, invertebrate frass, and also immature fungi. So at first glance, some of these will quite readily look like they're black fungi. However, if you're not sure, all of the above are quite amorphous in shape. There's no structure to them. But just check under the microscope. What you're looking for are spores or spore-producing structures, and none of the above will have those features. Okay, so I think it will be quite good now to start looking at the fungal groups that contribute to the group black things on sticks and have a look at the terminology. Okay. Although most of these fungi are in Ascomycota, they are not a distinct single group of very closely related fungi. Being a member of the Black Things on Sticks group is open to any that fit the criteria of being lignicolous, xerophilic, melanised, long-lived with many fruit bodies. So the above features have evolved from within more than one fungus group. And now that poses us with a bit of a dilemma, and that is because the terminology used for identifying these fungi will vary according to which group you've collected. And this can make identifying a member of this group quite a challenge. So I'm going to now explore some of the key terms used, but not all of them. So this is just a taster of some of the key terms that we use. One thing to say is you don't need to jot any of the terms down that we're about to talk about. This talk is being recorded, and you should be able to access it on YouTube soon afterwards. Okay, so for the terminology, I'm going to use this book. This is Microfungi on Land Plants. It's an excellent book to get started with. It's very comprehensive. It is, however, out of print, but you can still get hold of it. Okay, what I'd like to do is just open the book up and look at the contents page of this book and just highlight two chapters. We have plurivorous wood and bark fungi. This is the fungi that can grow on any wood from any tree. And then we have fungi specific to trees, shrubs and woody climbers. So these tend to be very host specific. Those two chapters you see there are very relevant to the fungi that we're looking at today. Just for information, if you're not familiar with this book, the section underneath covers a whole host of different areas: herbaceous plants, grasses, ferns, mosses, but we're not going to worry about that section, just the two top chapters. So I've found something on ash, and what I've done is I've gone to the section in the book for ash, and I can see here fraxinus, ash. There's a section there I can see on leaves. Well, I've got something on wood, so I'm going to turn the page and find the wood and bark section. So this is where I'm going to attempt to identify it. However, we're now going to start to see some difficult terminology. So I'm going to go through various pages. I'm going to start pulling out the technical terms and create a list. So just bear with me whilst I gather up the terms. The first one is Ascomycetes and Apothecia. Turn the page. Other Ascomycetes, Stromata, Perithecia, Ascospores. Turn the page for some more. I've got Hystrathecia, Pseudothecia, Hyphomycetes, Canidia, Coelomycetes, and Pycnidia. So those are the terms that I'm going to explore. I am now going to gather them all up, and you can see them all collected on the left-hand side. So what I'm going to do is to go through these words one by one, and we're going to understand what they mean. But just before I do that, I just want to explain how fungi can reproduce, particularly Ascomycota. There's two ways they can reproduce: sexually and asexually. So when the Ascomycota reproduce sexually, what they do is they have their spores inside an ascus. It's the long tube that we talked about earlier, and that is on the left-hand side. What can sometimes happen, they can reproduce asexually, and you'll know this because when you've collected your fungus and you put it under the microscope, you can often find lots and lots of spores but no asci. There's none of the tubes. You may find some of the spores are on the end of stalks, and this is the asexual state. A lot of spores but no asci, and the spores are produced either on the tips of or by the breakup of hyphae. The importance of understanding the sexual and asexual state is because if we come back to the list of words on the left-hand side, I can now split the list of words into two. The top list are specific to the sexual state, and the bottom list is specific to the asexual state. So if you find asci in your Ascomycota, you know you're going to be dealing with the top words in the book. If you find lots and lots of spores and you've got the asexual state, you know you're going to be dealing with the list of words at the bottom. Okay, what I'd like to do now is to start going through these words. So the first two I've pulled across are ascospores and conidia. So spores produced in an ascus, we refer to them as ascospores. Spores that are produced asexually outside of an ascus, because they're produced on the tips of hyphae or the breakup of hyphae, we can use the term conidia. So when you start to use the books, you'll see these two words. Okay, let's take the next set of words. So I've pulled across here from the sexual list Ascomycetes and apothecia. So this is a fruit body that is disc shaped, hence Ascomycetes, because they're disc shaped, and the spore producing layer is exposed on the upper surface. So this disc shaped fruit body is referred to as an apothecia. The best way to understand what this looks like is to imagine a jam tart. So the pastry cup will be the structure of the disc, but the nice jam layer is the fertile layer that produces the spores. So this is a jam tart, and this is similar to what an apothecia is structured. I'll give you an example for real. Here we are. This is Lansia, and it's on sweet chestnuts, and you can see the three discs, almost like a jam tart structure with an exposed fertile layer on the upper surface. What I have to say about this group, and the reason I've highlighted it, it is very, very unusual to come across Ascomycetes that fit our criteria of growing on wood, being melanised, and able to cope with desiccation, xerophilic. So for this reason, I'm not actually going to explore this group anymore. I just wanted to eliminate them from the chapters that we just looked at. I would, however, I'd like to introduce you to one that does fit this criteria, and that is Pyrenomycetes. It is able to do that, but this is very unusual. So like I said, I'm not going to cover these anymore. That means we go back over to our list of sexual words, and I've pulled these ones across. So these are other Ascomycetes that are not Ascomycetes. And these produce a fertile layer enclosed within a fruit body. So the asci are not exposed. Unlike the apothecia, where they're exposed on the upper surface, these are enclosed. I want you to imagine a jam donut. And here, all you can see is the dough-based structure. There is no jam because the jam is hidden inside. And this is a similar concept to those three at the top. To see the jam, what you need to do is to cut open the donut, take a look inside, and there it is, sat inside. And this is exactly the same with perithecia, pseudothecia, and hysterothecia. What you need to do is to cut them up to have a look inside. So let's go through and explore each of the three above. So I've pulled down perithecium. This is singular. This is what we're going to have a look at first. There is a hole at the top for the spores to come out, but what we cannot see is the fertile layer. That is because it is inside the perithecium. What we need to do is we need to cut it open, have a look inside, and I can now see asci inside containing the spores. Here's a photograph of one I've sliced open. What you can see is the round perithecium cut vertically down, the hole at the top for the spores to come out, and you can just make out the black spores in long lines within the perithecia. If I take one of the ascus out, I can see that it's single-walled. So the 8 spores in a long line are contained in a single-walled structure, and we call this unitunicate or one coat. You might think, what's the relevance of this? That will soon become apparent. Okay, so this group that produced perithecia with a single-walled ascus used to be known as the Pyrenomycetes. the burnt fungi. It's no longer a valid term, however, it's still commonly used because it is so well known. So that's perithecia done, and I've just put a green tick at the top. What I'd like to do now is to move on to pseudothecia. So there we are, pseudothecium, and it has a hole at the top for the spores to come out. If you are thinking that looks exactly the same as a perithecia, you would be quite right. Let's open it up and take a look inside. I can see the asci with the spores in, ready to shoot the spores out through the hole in the top. And what I'm going to do is to take one of the asci out. And there we are, there's the ascus. But here is a really important difference. The ascus isn't single walled, it appears to have a double wall. So you can see either 2 layers or one very thick layer. And the term we use for this is bitunicate or 2-coat. In reality, it's a lot more complicated than this. This is used for what you see down the microscope. So this ascus appears double walled, bitunicate, and it is this feature that you see down the microscope that separates pseudothecium from a perithecium. In the photo on the right hand side, the arrow is pointed out, and hopefully you can see that the wall appears very thick or has 2 layers. So that is pseudothecia. What I'd like to do now is to move on to Hystrathecia. And I'm going to bring a Hystrathecium up on the screen. This is the view from above. And what you can see is this long line on the top. And that is where the spores come out. And it gives it the appearance of a pair of black lips. And a view from the side underneath, it's very long and shallow. If I open it up, what you can see are the asci all lined up, ready to shoot their spores out through the slit on the top. And here on the right hand side is what they look like for real, attached to a piece of wood. So the slit on the top for the spores to come out. So there we go. We've just talked through perithecia, pseudothecia and hystrathecia and the difference between them, even though all of them have the fertile layer enclosed within the fruit body. So I'm going to pull across the next word now, and this is stromata. This is the last of our words from all of the sexual terms that we had on the left-hand side. So what is a stromata? It is fungal packaging that surrounds and unites several of the fruit bodies all together. Now, stromata isn't unique to any particular fungus group. So when you get a stromatic fungus, you might find within it perithecia or pseudothecia, and it can be found in some asexual fungi too. So what I'm going to do now for you is to take you through 5 common variations of stromata plus one that might catch you out. And at this point, can I just remind you not to forget to keep counting the fungi that you know of, and I'll flag later when you can submit in your totals. So let's have a look at the 5 common variations. Number 1. Let's just call these perithecia for now. They're lined up on the wood. They're all separate and isolated. And this is non-stromatic because it doesn't have any packing tissue uniting them together. So that's the first one. This is non-stromatic. And here's an example on the right hand side. You can see there the perithecia are scattered all across the wood. They're not united or joined up in any way, so they are all separate. So that is non-stromatic. Number 2, you can see here the perithecia are buried in the packing tissue. It unites them all together and they are spread out in one large layer. And we call this effuse stromatic. And on the right hand side, you can see an example of an effuse stromatic fungus. You can just make out where the perithecia are. You can just see the tops, but they're all packed in together into one stroma. Number 3, again, you can see the perithecia within a stroma, but these are now in discrete little groups on top of the wood. So this is discrete stromatic. And on the right hand side, you can see an example there of a discrete stromatic fungus. These are hemispherical mounds. And if you look very closely at any one of them, you should see lots of little black dots. And those little black dots are is the hole through which the spores are shot out of. Okay, moving on, we've got number 4. Again, perithecia within a stroma, but this time the stroma is immersed into the wood, and we call this immersed discrete stromatic. I'll bring an example here on the left hand side. You can see this fungus has formed discrete round stromas, and if you look very closely you can see little black dots in the center of all of the discs. That's where the spores are fired out from, and here the stroma has pushed the bark back just slightly as it's emerged out of the wood. Our final one, number 5. Oh, apologies. Yes, there's a photograph of one of the discrete stromas that I've sliced the top off, and you can see the perithecia buried inside. Number 5, coming back to that one, many, if not all of you, should be familiar with this one. This is a stroma that is erect and often stalked, and the perithecia are buried within it. One of the most, the group that are most well known for this are these. So this is dead man's fingers. You can see this is quite an upright stroma, and if you was to take one of these from this group and slice it open like I've done with this one just here, you can see just underneath the surface all the perithecia are buried, and they point their holes to the outside world ready to shoot their spores out of. So that is stromatic, erect and often stalked. So that's the 5 stromas. I did mention earlier that there would be one catch out that's not a stroma, and this is what we call a subiculum, and this is a fuzzy carpet under the perithecia. When you first encounter these, people do think, oh, this is stromatic, and it's not. It's, if you look carefully, you can tell them apart. So to explain this, little bit of a recap. Number 1, this is non-stromatic, so the perithecia is sat on wood. Number 2, this is the perithecia, they're sat in the stroma. This is stromatic on the wood, you can just see the top. But when it comes to a subiculum, the perithecia are sat on a fuzzy carpet underneath. And what I'm going to do is I'll bring up a couple of photos so you can see. You can see with this Rosalinia, you can make out the perithecia. They're quite distinct, and they're sat on top of a brown subiculum. Another very common fungus that produces a subiculum is this one. You can see the dark grey perithecia sat on top of a almost black felty surface, and that is also a subiculum. So there we go, 5 different types of stroma and 1 subiculum. Right then, so we're back to our list of sexual terms. If you look at the left hand side, and we've completed all of the ones on the sexual list, and we're back at this book, Microfungi and Land Plants. If you remember, I said this book is out of print, and you might be thinking, well, I might not be able to get hold of an out of print book. So what I'd like to demonstrate right now is how the terms we've just gone through are relevant in print books. So this is Fungi of Temperate Europe, Volume 2. This is the volume that has the Ascomycota in it. It's currently in print and a very popular book to use. I'm going to open it up, and I'm just going to take you to one of the pages, one of the identification wheels that deal with Ascomycota. And I've pulled across this list that we've just explored from the previous book. I'm going to try and match those words up with this book just here. So the first word is Pyrenomycetes, and you can see at the top of the book, Pyrenomycetes fungi. Perithecia is just there, marked in red. Pseudothecia and Hystrothecia are used on the opposite page over there. Stromata is just down the bottom there, and non-stromatic are used at the top just here. So understanding the terms from the previous book, although it's out of print, is still relevant today because the terms are still used in existing in-print books. Right, back to our list. So what I'm going to do now is have a look at the asexual words, those at the bottom just there. Okay, just to remind you, these fungi produce in two different ways. Sexual, the spores are in an ascus, in a long tube. If you see the asci, you know you're dealing with sexual words in the sexual state. If you find one of these fungi and when you have a look inside or you examine it, there's lots and lots of spores, but no asci, we are dealing with the asexual state and conidia. So let's go through and have a look at those words at the bottom. And the first one I'm going to start with is this, Hyphomycetes. So Hyphomycetes produce conidia, spores, exposed on hyphae, usually on stalks. And in mass, Hyphomycetes have a very felty looking appearance, and they look felty because it's the spores and the stalks that give it that felty look. And they can come in two forms. The first one is where the spores on stalks are effuse and spread all across the wood. And that gives the felty look, as you can see it in that holly twig just there. So that black feature on the wood is predominantly spores, and I've got an example of its spore on the right hand side just there. Very large, long spores. Another example of an effuse hyphomycete is this one here. If any of you are gardeners and you've had bamboo sticks stuck in the ground or stuck in a damp flower pot, you get the black sooty stuff on it and it comes off on your hands, and it's very felty looking, and it's this fungus. It's very fond of bamboo sticks. So this is also an effuse hyphomycete. Now, I did say they come in two forms. This is the second one. Instead of effuse and spread out all over the wood, the hyphomycete forms discrete little mounds. And here's a photograph of one. This is on a birch twig, and you can see the little round bobbles. At first glance, you might think, could that be a perithecia or a pseudothecia? When you take it off the wood and you bash it up a little bit and look at it under the microscope, you don't find any asci, you find no structure, you just find lots and lots of spores. And right in the middle of the photo on the right hand side, you can see a stalk with the spores still attached. So lots of spores and no asci. So you know you have an asexual fungus, and this is a hyphomycete. If you want to know more about hyphomycetes, there is a BMS talk by Marcus Yeo, and it is available on YouTube. I would suggest going and watching it. It's very, very, very interesting and useful. Right then, our next word, coelomycetes. Coelomycetes are the complete opposite to what we've just talked about. So instead of the conidia, the spores produced openly and exposed, here, the conidia are enclosed within a structure that looks like a perithecia, yet you cannot find any asci. There's just lots and lots of spores inside the structure. So this is enclosed. So here we go. I brought one up on the screen. We call these pycnidia because you can't find any asci whatsoever. There's a hole at the top for the conidia to come out. Yet when you open up and have a look inside, all you can see are a lot of spores, and maybe spores on stalks. Here's a very common example. This is on holly, a holly stick. This also grows on holly leaves. It's black, and the hole has this little white fringe. And if you cut one of those open and have a look, you will just simply find lots of conidia. Here's another example. The pycnidia I'm going to circle just so that you can see where it is. There's the pycnidia. I've squished it under a cover slip on my microscope, and the conidia have come streaming out like smoke, but I can't see any asci. So this tells me this is a pycnidia of a coelomycete. Okay, so we've just done a load of technical terms for the asexual state, and I just want to show you how relevant those terms are in an in-print book. So I've come back to this one, Fungi of Temperate Europe. I'm going to open it up, and this book does have a section specifically for asexual fungi. Here's the terms that we've just been discussing. And let's go through and have a look. So we're talking about the asexual state, and there it is at the top of the book. The conidia is mentioned in the text. Hyphomycetes is mentioned across there, Hyphomycetes fungi, and they produce conidia externally. So that is mentioned just alongside the title. Coelomycetes that we've just talked about produce their conidia internally. So there's Coelomycetes fungi, and the conidia produced internally is mentioned just down there. So you can see quite easily how these terms are relevant in imprint books. Okay, that's our list of technical terms done. There are so many more I'd love to take you through. However, we would be here all night. So let's leave that for now, for another time. So where are we at right now? So I've covered the first 5 areas that I wanted to talk you through. What we're going to do right now is to key out a fungus for real. And we're going to use those terms, and we're going to use a book. So let's key out a fungus. Right then. On the left-hand side, what I'm going to do is to put my summary of my findings as I research my fungus and the investigation itself. So what I'm looking at and what I'm analysing will be on the right-hand side. So let's go through and have a look. Here we go. So I've gone for a walk in the woods. I'm stood under a sycamore, Acer pseudoplatanus. I've got some fallen leaves. There's a moss-covered piece of wood on the ground, and there's something black sticking up out of the wood. It's a melanised something or other. I'm not quite sure if it's even a fungus, but it could be stromatic. So I'm going to take the key elements of that and just list it on the left-hand side. So we are on wood, Acer pseudoplatanus, and it's upright. Okay, I collect it. I'm now going to analyse it a little bit more. I can see it's 4 centimetres tall. It's got a stalk. It's quite matte and smooth, maybe slightly scaly. I cut it open. It's white inside, which is surprising, and it's got a dark edge. So I'm going to look even more closely at that edge, and I can see these round spheres buried just under the surface. So I'm thinking to myself now, this could be Pseudothecia, Perithecia, or Pycnidia. I just don't know, but I suspect it might be fungal. So I'm going to capture that information and list that across. Now, what I'm going to do is take a small sample of the jelly-like interior from within one of the spheres. And I'm hoping to see some fungal matter, because that will help me identify what this is. So there we are. Lots of dark coloured spores in Asci. So this is definitely not a Pycnidia. I can see the spores in Asci. And I'm going to take a closer look. Oh, I'm just going to update my list. So I now know that this is stromatic with Perithecia or Pseudothecia and dark coloured spores. I'm going to take a closer look at an ascus, and I can see here that the spores are pushing right up against the single wall of the ascus. So this tells me this is Unitunica. It's not Bitunica, and it's from a Perithecia. So let's update my summary. And I'm now going to zoom right in and look really closely at the spores here. They're very dark, but I can see a germ slit. And that's how these spores germinate. The hyphae will come out through that germ slit. So I'm going to update the information on my summary on the left-hand side. So there we are. I've done my research. So now I need my book to try and key this fungus out. So from investigation, I'm now going to go to identification, and I am going to use this book, Microfungi on Land Plants. Let's open it up and have a look at the sections. Well, it's going to be on wood, so I have a choice of plurivorous wood and bark fungi, or fungi specific to trees, shrubs and woody climbers. It could be in either of those. So I'm going to just pick one at random. I'm going to go for fungi specific to trees, shrubs and woody climbers. And as I know it's on Acer, I'm going to go to Acer within this book. Let's have a look. There we are on Acer. But if you look just underneath Acer, it says on leaves. Well, this is most definitely not on leaves. So I need to turn and find the section for wood and bark. And there it is, just at the top on wood and bark. So let's go through all the technical terms I'm presented with and see if I can identify this fungus. So the first term I've got at the top is Ascomycetes. The spores were not exposed on a fertile layer on the top like a jam tart. The asci with the spores in were enclosed within a perithecia. So it's definitely not Ascomycetes. It's going to be under other Ascomycetes down at the bottom just here. Here, I've now got a key. So I'm going to start working my way through this key. And I've got two options. Ascocarp. So this is a new term, and it's just a structure that contains asci. And it's asking me, is it setose with dark seta arising from a subiculum? In other words, does it look hairy itself or sat on a velvety carpet? Or is it without seta, without hairs? Well, I didn't see any hairs. So that means I am going to go to number 3 in the key. And here it's asking me, is the ascocarp, the fruit bodies, immersed in a large, erect, club-shaped stomata? Is the ascocarp immersed in round, reddish-brown superficial stomata or not? Well, I've definitely got a stomata, so it's one of the first two. And mine's not reddish-brown and round, it is large, erect, and club-shaped. So that means it's going to be the top one, and that means I need to go to number 4. So let's go to number 4, which is on the next page. Step 4, and let's see what that says. Two options: brown ascospores 12 to 15, brown ascospores 20 to 31. Well, if I look at my summary, they were 15 long, which nice and neatly says it's the top one, which is Xylaria longipes. Excellent. So let's have a look at the description for this fungus, Xylaria longipes. Perithecia immersed in a swollen dark brown stalk stroma, tick. Ascospores brown with a germ slit, definitely a tick. On rotten fallen branches of Acer pseudoplatanus, tick. Yeah, I wouldn't lick this. Right. Now, it says at the top there, Fig 3 to 8. Let's go and get the picture up and compare the picture from fungi, microfungi and land plants to the specimen that I've got. And I think that's a pretty good match. Yeah, I'm happy with that. Okay, so keying out a fungus. Right, so we've got two sections left to go through. So number 7, some common species as well as some highlights. So let's go through and have a look. So I've got 7 commonly found black things on sticks for you. And this first one is Pyrenomycetes scaparia. This has perithecia buried within the wood and long necks that come up out of the wood, and from there it fires its spores out, and the long necks coming out gives the wood the spiny appearance that you can see in the top photo. This is quite common and really nice to find. Number 2 is Eutypa flavivirens. This has perithecia buried in an effuse stroma, but notice that this Eutypa produces quite a disorganized effuse stroma. It's not nice and neat. It looks like it's broken up and a bit crazy. And the nice thing about this fungus is when you scratch the surface, it is sulfur yellow inside. It's really, really nice. And what I've highlighted in this photo for you are a couple of perithecia buried in that lovely sulfur yellow interior. This is often overgrown by another fungus called Capronia, and we'll be coming back to that shortly. Lepidostoma. This is, once you get your eye in, is quite easy to find in the field because it turns the beech bark a reddish colour, which contrasts with the surrounding wood. The perithecia are buried as you can see in the top right photo, in little groups underneath the bark, and the holes come together through a tiny black pimple coming out of the wood, and they produce coffee bean spores, is what I call them. The next one is Hypoxylon petronae. This is very common on ash. It's perithecia in a few stroma with a distinct black margin, and I particularly like the photo on the right hand side because the stroma has followed the ring growths of the ash tree. This is candle snuff, and this produces perithecia. However, this has two very distinct stages. So the first stage of candle snuff, the asexual state with conidia, is this one. So whenever you find candle snuff in the field with the white top, the white is produced from or by the conidia, which is where sometimes when you flick it or you knock it, a white powder comes off. This is not in the mature sexual state. Candle snuff is only mature when the upper part goes completely black and the perithecia form, and it becomes quite knobbly. And I've circled just a couple on there where you can see for some of them the little hole where the spores will come out of. Okay, this one, Arthrinium, this is the one I talked about earlier, grows on bamboo sticks. So even if you've got just a balcony, put some bamboo sticks in a pot of moist compost and be patient. This usually turns up. Very common, and I do like the spores because they look a bit like donuts, which you can see in the bottom right hand corner. This is a coelomycete. This is Phasidium. We've talked about this earlier, and if you cut these open you should find lots of conidia inside. Right, so that was the common ones. I've got 7 unusual or rare fungi to have a look out for, and I'll explain why some of them are rare. This one is Hysterographium fraxinii, and in some of the books published a few decades ago, this has been described as extremely rare or uncommon, except in parts of Wales. I now find it very frequently, and it tends to grow on dead ash twigs. Whether there's a link between its distribution now and ash dieback, I don't know, but it's certainly a lot more common than it used to be. It's a hysterothecium, so it's got the slit on the top for the spores to come out, and very large spores with lots of divisions inside, as you can see in the photo. This is a Glyphium elatum, or hatchet head. I particularly like this because it lives up to its name of having a hysterothecium that's elongated like a hatchet head and really long spores. The next one is Hypoxylon phascoides. For some of you, if you think back, there was the Lost and Found fungi project about 5 or 6 years ago that were looking for some of our rarest fungi, and this was one of the ones listed. The challenge with this is it grows on alder, and so do a couple of other hypoxylons, and this might be why it went under the radar for so long. When you get to know this really well, it is smaller. It's the smallest hypoxylon that grows on alder in size, but the surefire way of separating it from the other common hypoxylon is to put the sample into potassium hydroxide, or KOH. So Hypoxylon phascoides releases a wine-coloured into the potassium hydroxide, and Hypoxylon fuscum produces this orangey brown colour. So that's a really easy way to tell the two apart. Something to look out for. Many of us will know King Alfred cakes, particularly further down south where it's far more common and it grows on ash, but there are several other Dordinias and they have a strong preference for burnt wood. There's a couple on the screen there, something to look out for. Whenever you come across a tree that's been burnt, have a look for Dordinias coming out of them, and they're very likely to be one of the other species. Right. Do you remember this? We talked about this earlier. This is your type of flavovirens, the lovely sulfur yellow interior of this black effuse fungus. Well, there are 2 fungi in this photo. We've got the type of flavovirens and we also have another fungus. And I'm just circling the fruit bodies right now. This is why this isn't often recorded, because it's really difficult to see. It's Capronia nigerrima, and the photo just there, you can see it looks a little bit like blackberries. I think it's a parasite. It is very common, but very easily overlooked. If you cut one of the stroma, this is a stromatic fungus. If you cut one of the stroma off and do a cross section, you can see all the pseudothecia form like a circle. And in the center there, you can see what was left, or a partial bit of the eutypa. Close look at the pseudothecia, and there we go. And it has very short hairs in the top. A really interesting fungus is this one. This is the angel share fungus, or Bordonia. I think it's probably more common in Scotland because it feeds on alcohol vapours. Therefore, distilleries and bonding warehouses is where you can find this. It's fantastic. It turns the sticks and the barks of tree completely black. You can do the same with anything in the vicinity of a bonding warehouse or a distillery. The hyphae are made up of these little circular cells that can break up to form conidia. So this is a hyphomycete. And I like the photo in the middle of the screen because the yellow brain fungus contrasts so well with the hyphomycete. Finally, this is a fungus I've not seen for this section. So this was found in the New Forest by Alfred. So hopefully Alfred is on the call. Thank you for sending this up. He sent me some specimens. I've yet to see it for real, but I'm for in the wild. But I'm very grateful for Alfred sending these up. This is like a candle snuff, but notice that it's very streaky, Xylaria cinerea, as it goes up. And I'd really like to see this. So maybe a trip down south at some point. So that was 7 rare fungi. What I'd like to do, the next section is when you get involved in this group of fungi, your eye wanders and you move away from sticks. And so I'm just going to touch on some of the other substrates that give us black things on other substrates other than sticks. This is going to be quite brief because I'm going to give you references that you can refer to find out more information. So, black things on other substrates. On mosses and liverworts. So there are many little black things that grow on mosses and liverworts. This is Epibryon, and if you want to find out more about this group, some of you may remember this wonderful talk by George. This is all about Bryophilus Ascomycetes. So if you want to find out more, have a look at that talk. It is available on YouTube. Dead leaves provide a whole host of things. Holly is particularly good. Microtherium ciliatum is on the screen now. It is not the large black dots. That is holly speckle. If you look very closely, there's these much smaller little grey dots, and it's this spectacular looking fungus that, in my opinion, looks like a cathedral rose window. Another thing that you can find on holly leaves is Pyrenomycetes elysis. They look like black sea urchins. You can just see them there on one of the spines on the holly leaf, and collect one of those off. This is a coelomycete, and there we go. On living leaves, herb robert is a nice example. This is Coloraria, and these are pseudothecia buried in the leaves. And I like this because the spores look like the soles of shoes. Okay, moving on. On lichens, I was waiting to be picked up to be taken on a foray, and I got very interested in this bin lid. So there is xanthoria growing on it, and unfortunately the cigarettes had damaged the xanthoria, but I did notice on the apothecia, the jam tarts, these little black dots, and when I collected those, you might recognize this, I saw the pycnidia and the conidia on this lichen. If you want to find about more about this group, there is another BMS talk by Faye on lignicolous fungi. Well worth having a look at that video, available on YouTube. On dead herbaceous stems, there's a whole host of stuff. I'm going to do this nice and quick. Periconia bisoides and Acrospermum compressum, both very common and easily found. On dung. Okay, so if you are pushed for time on a foray and you collect some dung, the great thing about this sort of stuff is that you can incubate it when you're ready, and it can produce a whole load of black fungi. And here I've brought Podospora tetraspora. I've collected the perithecia, given it a bit of a squish, and you can see that the asci have all burst out. This is a really, really good thing to get involved in. And if you want to find out more, there's a wonderful talk by Tony Leach on coprophilous fungi. Okay, so that's me to the end of all of the species and the different highlights. I've got one section left to go, and that's the resources for study and challenges. Before I take you through that final section, there's one thing I'd like to ask you to do, and it's this. Remember, I said how many of the 38 named fungi did you know? And I asked if you could collate that up. If you could fire your results into the chat now and let us know. Remember, there is a prize for the highest, a stick with a black fungus on it. So there we go. Right, so if you could send that into the chat, that would be lovely. And whilst you do that, I will wrap this talk up with resources for study and challenges. Okay, resources you need. So I've split this into two. Resources you need out in the field when you're collecting, and then back in the lab. So out collecting, take photos. Really useful to take photos. That ensures you capture more information than you might think you need, but useful for when you get back to your lab. Hand lens to sort out the amorphous stuff from the fungi. A penknife to cut small samples, and a fold up pruning saw. Absolutely invaluable is a fold up pruning saw. And then you want a basket to carry the large sticks and small plastic tubs to store small samples. Bear in mind, I've just told you to carry a penknife and a pruning saw. You either need gloves or plasters. When I first started out, I needed lots of plasters. I've come wise to the whole thing, and now I use gloves. When you get back into the lab, books. So I've recommended these two books. There are other books available, but you know what? Microfungi and Land Plants is a really good book. It's very comprehensive, and Fungi of Temperate Europe does have a lot of good stuff in it. Fungus glossary. If you're starting out on this and you're quite new, photograph the glossary or try and print it out, and then it makes things a lot easier for you. Microscope with slides and cover slips. And chemicals. To be really honest, water, when it comes to microscopy, is the best chemical going. It's pretty much the only one you need. However, you might need at some point to use Lagol's solution, which is iodine. Meltzer's is almost as good as Lagol's. Okay, make sketches of the key features as you see them down the microscope, and compare your images that you see down the microscope with the images in the back of the books, particularly with microfungi and land plants. It's really good. And use online resources such as the images option, and then compare what you're looking at to what's online. And the last one for resources you need is patience. What I would advise here is worry less about getting to an ID and learn about the fungal features first. Take your time and learn your features and learn your terminology. Okay, the very last bit I'm going to talk through now are the challenges, and I've split this up into when you're collecting and back in the lab. So challenges when you're out collecting far more specimens than you can ID. One stick could provide you with quite a few species, so don't over collect and don't overload yourself. Conversely though, collecting something with only 1 or 2 fruit bodies might not be enough. If you're practicing and learning, you want to get a specimen with quite a few fruit bodies so you can practice. Okay, overexcited dogs. So you've just found yourself a nice stick and a dog sees you, I'll run out the bushes and there's a bit of a fight between you and the stick. So watch out for overexcited dogs. And also people putting sticks in your basket when your back is turned. I find this extremely annoying because I get back and I can't understand why I've got all these sticks in my baskets with nothing on. So let's do the last little list. Challenges. Getting too focused on an ID, otherwise you failed. Just learn the structures, give yourself 30 minutes and then move on to something else. Perithecia are really quite hard, so when you give them a good whack with a cover slip on, they might break. So break up your perithecia first or remove the contents and then cover slip on top. Historically, the naming of Ascomycota was very complex. Many have two names. You will encounter this. Don't worry about getting the most up-to-date name. If you get to an ID, whether the name is up to date or not, that's perfectly okay. And finally, new species not listed, because not many people do this group. There's lots of new discoveries to be made out there. Right then, this brings me almost to the end. Quick thank yous for Dr. Joe Taylor for reviewing my slides, Becky Yar and the UK lichenologists for the jam tart inspiration, Gracie the dog and Sally Hawkes, and finally to all of you for listening to me talk about one of my favourite subjects. So yeah, thank you very much indeed.
Well, thank you so much, Cameron. That was absolutely magnificent. I know so much more than I did before. We have a clear winner. So Carolyn Hargest found 28 species. But
Well
we
done, Carolyn.
have several more. We have Andy, I don't have a surname, who found 24. And Carol Hobart found 22. And Steve Pennington, 21. Sarah Ebden found 15. So those are pretty good numbers. I only found 5. We haven't really had any questions, so I'm hoping that we've had a couple people saying how much they enjoyed it. That was fantastic. Thank you very much. Very interesting. I just have a question, being very much a beginner. You said when you flicked the Xylarian hypoxylone, the candle snuff, the white ones, that you can see the smoke coming out. What is that then? If it's not
so that's the conidia. That's the asexual spores. That's the white. So when you give it a flick, it releases all of the conidia into the air.
Okay, so that's not okay, right? Because I always thought that was the spores coming out, but it's the conidia, the asexual spores.
It is. It is.
Okay. How well studied are the Xylariaceae on dung?
I'd reasonably well. I think there's a lot of probably change coming with more DNA work on them, but I know there are a couple of really good dung fungi experts out there, coprophilus experts that have done a lot of work. But I think molecular work might start to reveal some unknown species out there.
Right. Okay. Thanks for a great talk, a great resource to return to when attempting the next black thing on a stick, is from Jassy. And is that tremella associated with the angel's share?
So there was a peniophora further along on that stick. So I know what I think that was Eric referring to that. Yes. So that tremella is a parasite on peniophora and it was on the same stick, but not in the photo.
So just coming out of the...
Yes.
Yeah. Okay. How would you break up the perithecia? How do you say that word again?
Perithecia.
Perithecia. Sorry, everybody. How would you break up the perithecia before trying a cover slip? How would you do that?
So if it's stromatic, it's sat in a stroma. I cut the top of the stroma off, so the perithecia are sat in place. So when I look down, I can see the jelly still sat in the perithecia with the asci in. I now use a very fine needle to scoop the jelly out and place it into a drop of water on my slide. If, however, the perithecia is loose, what I do is I'll put it onto my microscope and then I'll crush it with a needle first, you need a bit of practice because hard parathecium will suddenly ping off and disappear. So you need a bit of practice, bit of water, and then crush it into the water and then put a cover slip on. It takes practice. You will get through cover slips.
Right. Okay. Okay. We have another question from Lydia. Do Hysterothecia have Bitunica asci?
They do. They do indeed.
Do you know of any key groups of black fungi on sticks which are cryptic, worth targeting for DNA sequencing? That's from Andy.
I think quite a few of them. So the hypoxylons, until very recently, there were very few species. Someone revisited them, and suddenly the number of species exploded just based on morphology. I think that if they start to do more molecular work, we'll find more. So the one on Alder, there seems to be a cryptic species in there somewhere, because when you extract the colours from them, there are other colours coming out, hinting there are other species. But there's no work done on them. So I think, yeah, the answer to that is there is. There is more to do there and more hidden.
Brilliant. Okay. Just a couple more compliments for you. Brilliantly structured talk. How fascinating. Thank you. I think I actually understood most of it.
I think I did too.
I agree. I feel like I know so much more. Thank you. That helps. Great talk. Okay, so Jassy says, do they tend to make spores only on fallen, dead wood, or can they grow on sticks still attached to plants?
Yeah, both. Yes. So there are some. There is a xylaria that only grows on attached ash branches at the top of ash trees. The moment the branch breaks, the fungus stops growing. And there are others that only grow once the branch has landed on the floor. So the answer is it depends on the type.
Mhm. Okay, well, we're just getting lots of excellence. I think you wowed everybody so much they have no
Oh, well,
questions.
thank you.
Anybody else? What do you better get some tree climbing kit? And
Uh-huh.
okay, Sarah says, you mentioned that you can ID dead black things on sticks. Are there any tips for this or just more persistence?
I would be persistent when you collect them. Collect enough to practice with, play around with the keys, and you soon learn. You soon learn the key features. What you start to do then is when you look at them at home and you key them out, you go back in the field and you find them again. You start to recognize the features you saw in the back at home, back out in the field. It doesn't take too long. It doesn't take too long.
Okay. Julie says, are they pathogenic?
Some of them are to other fungi. Some of them are, yes. Many of them are not. Some of them are lignicolous, but it's an artificial group, what I've just gone through. But yeah, some of them are parasites.
Okay. We've got another question from Alfred who says, have you got any idea why it might be so hard to cultivate Xylariaceae on dung to get them to mature?
I don't know. I don't know. And I know why Alfred has asked that question. It's a really good question, because if I knew the answer, there's a couple that I'd like to get to mature. I'm not sure, actually. I'm not sure why they're not doing that.
Okay. Anyone else have any questions? Okay, I've collected several black things on hogweed. Do these guidelines apply?
Do you know what? I'd say yes. If it means you can look at them, I'd say yes. Yeah. Black things on herbaceous stems is covered by microfungi and land plants, so yes.
Okay, great. I have to say that I found your keying really fascinating, because that's one of the things I've got trouble with. And it was lovely to see you go through each step. And, you know, I mean, it may have seemed obvious to a lot of other people, but it really, really helped me a lot. Okay, Sal says that he loves your background models, Cam.
Thank you.
Right, well, if nobody else has any questions, brilliant talk. Thank you so much. Really inspiring. Excellent. Everybody is really, who's come to the talk is really happy with it, I'd say.
Well, I'm just really grateful that people listened and let me talk about a favourite subject. Thank you.
I also think the fact that it's been recorded, because we could really get those terms in our minds, and your drawings were just really excellent to explain what every single part was. I thought that was great. Oh, here's another question. Has the prevalence changed with climate?
I think there's an impact there that we might not fully appreciate just yet. Like I touched on it very briefly, that there's that fungus that grows on dead ash twigs, listed as very rare in the books, and I'm finding it everywhere. Is that as a direct result of ash dieback? I don't know, and that's quite obvious. So what the climate change impact might be, I'm not so sure, but yes, I don't know on that one. It doesn't help that I've moved quite considerably within the UK, so I live in a very different climate to what I was used to, but I think there will be consequences of that.
Right. Well, I think all of us who have listened to your talk will now be going out and collecting fungi with a new viewpoint. I mean, I have been interested in black things on sticks for a while, so I was thrilled to be asked to share this. But I now really do feel I know a lot more about it. A thoroughly enjoyable and inspiring talk.
Thank you.
That was from Chris Ansley.